In this video, we'll be taking a second to appreciate this piece of art. Actually, we'll be looking at karyotyping and chromosomes. So these two topics go hand in hand. So let's just get straight into it. When we look at a human being, so once again, the rock as an example, when we look very closely at him, what is the smallest unit of life that we see? We all know that smallest unit of life is a cell. We go very close, we'll see cells, right? We are made up of billions and billions of cells. Inside those cells, we have many organelles, right? Organelles are like workshops. They each do a different job for our body. Each have a different function to the cell. And all of our different cells have different functions. We have muscle cells that contract. We have brain cells that think. All of these different cells have different jobs, right? Now, when we look in this part, what is this part called? This part is called the nucleus. Inside the nucleus, we have the code, the instructions of what our cells are meant to do. This is called DNA, right? And our DNA is organized into something called chromosomes, right? So let me show you here. It's organized into chromosomes, many of these. So if we look closely, we can see that we have many of these chromosomes. So we don't just have one. Specifically, humans have 46 chromosomes. Now, scientists, what they normally do is they take out these, if they will isolate a cell like this, and they will organize these chromosomes in terms of size. So kind of like this. So this is what I mean. So they will take out all those 46 chromosomes, go from biggest to smallest, and then this extra piece here, which I'll talk about soon. And this, what they call this, is a karyogram. Okay, so let me organize this. A karyogram. Karyo means nucleus. Okay, so a karyogram is like a nucleus diagram. It shows all the chromosomes in your nucleus. So that's what it stands for. So we immediately know that this is a human because we can see 22 plus 23 plus 1, that's 23. So we have 23 pairs of chromosomes, so that is 46 individual chromosomes. Okay, so we know this is human. Now, there's another word that is a bit confusing, so we need to distinguish it real quick. It's called karyotype. So karyogram is a picture of your chromosomes, whereas karyotype is what this picture is saying. Okay, so a karyotype would be the number and types of chromosomes present in a nucleus. Never mind. Okay, so that's the difference. Karyogram is the picture. Karyotype is what's going on in the picture. So I would say this is a picture, this is a karyogram, and then I would say the karyotype is human because there's 46 chromosomes. In this case, this human is female, and this human is normal, supposedly. There is no extra or missing chromosomes. There's always 2, 2, 2, and so on. Okay, so that's the main difference between karyotype and karyogram. Okay. Now, one more thing. What can we notice from a karyogram? Like I just said, you can notice if there's extra or missing chromosomes, and that would normally lead to disease. Or you can also tell if it's male or female. If you don't know, males have one X and one Y chromosome, so we are XY. Females have two X chromosomes, so they would have X and X. So in this case, we know this DNA was taken from a male cell, because there's X and Y. Y is normally the smaller chromosome, whereas X is much larger. So, if we had a female, it would look like this. It would have two X chromosomes, X and X. This would be a female. Okay, and normally the male and female chromosomes are always in the right bottom corner, just like this. They will never be up here or down here. They're always in the right bottom corner. So as soon as you get a question, is this male or female, always look at the right bottom corner and you will see the chromosome. Even if the X and Y they're not showing you that, you can tell because the X's are big and the Y's are small. There's no such thing as Y and Y. That doesn't exist. Males are X and Y and females are X and X. So here, real quick, just to make it clear, this is a karyogram. Now, what is so first, before I explain these two words, can you think of an example of a karyogram or a karyotype that would be different due to a disease? So one that you need to know is trisomy 21. So look at this real quick, this karyogram, and tell me what's different from the normal karyogram. If you notice, you'll see it's decreasing in size as they go on until 22. It's still decreasing, so it's still organized well. One thing you'll notice is the fact that on chromosome 21, there's 3. There's not 2, but 3. So that's why it's called tri. Tri means 3. So it's called trisomy 21 because it occurs on the 21st chromosome. When this happens, it's called Down syndrome. The physical characteristics that you get is these two. So you have these, your eyes will normally be smaller and slanted. They'll be slanted differently, right? So you can see the edge points more up and the eyes are slightly smaller. They normally have a larger forehead. So there's various characteristics that you can see. Sometimes when you go out and you see somebody with Down syndrome, it's quite easy to recognize because they have special features, right? And another one that's 50-50. So 50% of people with Down syndrome have this, you know how on your hands you can see lines, right? So in 50% of people with Down syndrome, this line is singular. You won't have 2 lines. You just have 1 line straight through. That's typical, okay? But again, it's not all the time. Like, not all Down syndrome people have this line. So that's what I mean. This is one example of how a karyogram is useful, because before a baby is born, scientists will isolate the DNA from that fetus and produce a karyogram, and from that they can see if all the chromosomes are normal, if the baby will have a Down syndrome, or if it's male or female. So in this case, again, this person with Down syndrome is female because it's got 2 X chromosomes. So in a nutshell, that's why karyograms are useful for 2 reasons: determining gender and determining if the baby will have a disease by will have a chromosomal disease, because if it has an extra or missing chromosome, it could have a disease, and so that might be noteworthy for a parent to know prior to the baby being born, so they can prepare for such a situation, right? So there's many other examples. Trisomy 21 is just one that you need to know. Now, next part, autosomes. So what do I mean by autosomes? Autosomes are all the chromosomes in a karyogram or in a cell that are non-sex chromosomes. Okay, so all of these have nothing to do with determining whether you are male or female. They are involved in every other characteristic, like your height, hair color, all that kind of things, all your other characteristics. Where sex chromosomes are these 2 in the right bottom hand corner. Okay, they determine whether you are male or female. All the rest, so they're called sex chromosomes, whereas all the remaining ones, so all the first 22 pairs, they are autosomes, determining all the other characteristics, non-sex chromosomes. Okay? Hopefully this is pretty much the main things you need to know about karyotyping. Now let's get on to the next part. How? So right here I mentioned what we can tell from a karyotype or a karyogram, right? How did they achieve this? How did they make this happen? Because I showed here they have a human and a cell, and then they make a karyotype. But what specifically? Okay, let me show you. Because this you need to know for some multiple choice questions. Here is a female, okay? Hard to understand this image, but I'm going to try and explain it. So this is her leg. Okay, she is lying down, so her leg is lying down like this. This way, this way is her upper body. So you can see here, her leg and her upper body is meeting, right? This here is the vagina, okay? So, and this here would be her butt. This would be the bottom. So she's really lying down facing up. Because I can tell this because this right here is her spinal cord bone. So this is her bone, her backbone. Her spinal cord, right? Spinal bone. So if you feel in the back, you can feel these bones, right? So that implies that this is her back. So if this is her vagina, if this woman is pregnant, and this in the front would be her bladder, and then this in the back here would be the anus, okay? So if we go into the vagina, we can see if she's pregnant, that she will have a baby in her uterus, right? You can see this one is a small baby. It's just recently started growing. And where the scientists get these cells from before a baby is born to be able to tell whether it has a syndrome or whether it's male or female is from two places. Right. Either the amniotic fluid of the fetus. So when the fetus is growing, we know it's growing in a sac-like fluid, right? You can see here. So here's a better image. Let me make this one bigger. It is growing in this fluid, and this fluid helps to protect the baby from when the mom is moving so it doesn't bash itself around everywhere. And at the same time, as this baby is growing, its skin cells are falling off into this fluid, and its hair cells and all other kinds of cells are falling off, right? So that means a scientist can pretty much insert a needle into this amniotic fluid and extract the fluid, and then inside they will find the baby cells, okay? And based on these cells, they can make a karyogram, okay? So that's one place, the amniotic fluid of the fetus, okay? Second one is blood sample. So that's, for example, if an adult or something like that wants to look if they have normal chromosomes or things like that, or if they have a specific disease. Then you just take a blood sample, because that's simple for an adult. Just for a baby, it's quite difficult because they're so small. Now the last one in a baby would be chronic, I spelled that wrong, Charionic villus sampling. So this is similar to amniotic fluid, but a different place. So the amniotic fluid would be surrounding the baby, whereas chorionic villus sampling would be over here in the placenta. These cells also are going to be the same cells that the baby has. Therefore, when we extract these cells from the chorionic villi of the placenta, it will be accurate to the baby's DNA. So the chorionic villus is just a place in the placenta, okay, which is connected to the baby. Therefore, the cells are the same, and therefore the DNA is the same. So two places: amniotic fluid, chronic villus, or blood sample if you're an adult. Now, once that cell is taken out or isolated like this, now the scientists will wait until the right phase of the cell cycle, because we know the cell, a typical cell has a typical cell cycle where the DNA is at first not condensed, so you can't really see it like in this picture, and then as the DNA is being condensed, it turns into prophase, right? Because prophase will be the phase where the DNA is supercoiled and it forms chromosomes like this. So you can see them really clearly. The nucleus will be very dark. So the scientists will essentially wait for this phase to happen, because at some phases you can't see the DNA and they're there, but they're not condensed, so it's no point taking them out. So the scientists will wait for prophase, where the DNA is really clear and in chromosome shape. Then they will take out that DNA of they will take out those chromosomes and organize them pretty much in size like this, and then that's how simple it is. Now you have a karyogram, and now the doctor can look at this karyogram and determine some basic things, not many things, but some basic things that are important. Now let's move on to this part. So if we look at an individual chromosomes, what are some basic things you need to know about chromosomes? So right here, we have what we call homologous chromosomes. Homologous means that this right here is a homologous chromosome, meaning two chromosomes connected. One chromosome, another chromosome connected. And they're called homologous. Homo means the same, because one is from the mom and one is from the dad, but both of these chromosomes have the same function. They may have slightly different properties, but they have the same function. That's why they're called homologous. So for example, let's say this one is your dad's and this one is your mom's. This part, the top part of these chromosomes, may each encode hair color. Your mom's hair color might be different from your dad's, but they still have the same function. So if we move down, here we find height, and here we find height of the dad, and then here we find height of the mom. It's still the same function, but they might have slightly different alleles or characteristics, like your mom might be short and your dad might be tall, or vice versa. So that's what homologous chromosomes mean, and that's what we see here on a karyotype, okay? So these are all homologous chromosomes. There's one from your mom, one from your dad, and they have the same function. That's what homologous chromosomes mean. Okay, now homologous chromosomes are diploid. That means diploid means di means two. That means you have two individual chromosomes connected together, one from the mom, one from the dad. So in a nutshell, it means both chromosomes of each homologous pair, meaning you have your mom's and your dad's. So that's what we see in a karyotype. That would be an homologous pair, and this would be a diploid DNA, meaning you have one from your mom and one from your dad for each chromosome. Where do we find these diploid DNA? You find it in your body cells. So a body cell is a cell that is not a gamete, meaning it's not a sex cell, meaning it's not a sperm or an egg cells, all the rest of your cells. So your skin cells, your muscle cells, brain cells, all of those cells except your sex cells, because your sex cells are not diploid. They're different, and we're going to get to that just now. So your sex cells are in fact haploid. Haploid sounds like half. So your sex cells would contain only half the DNA. So they would not have one from your mom or your dad. It would be one chromosome that is your DNA. So a mix of your mom and your dad's. So let me show you. So instead of looking like this is a diploid diagram. Instead of looking like this, if you look at your sex cells, so your sperm and your egg, they would be half. So you'd only have one of this one, and one of this two, one of this three. So it'd be exactly half. Diploid is when there's two of each. Haploid is when there's one of each. And that makes sense because when you are passing on your DNA, it's from your sperm cells. And if your sperm cell combines with a woman's egg cell, if you each have half or haploid, that means when they combine, you will form diploid, which is what your normal skin cells are like, right? That'd be weird if you had diploid and they combined. That would form like a tetraploid or something, right? That'd be crazy. So it makes sense that your DNA or your gametes, so I have to make this clear, your gametes, which are sex cells, science name for sex cells, so egg and sperm. So it makes sense that your egg and your sperm are haploid, since when they combine eventually, that will form a diploid, which is what your somatic cells are made of, all your body cells. Okay, last thing here, that's very important. When you look at this image here, which organism do you think has the most DNA? Most people, including me before, would say this guy, the rock, right? And then probably this fish, and then this flower, and then this bacteria. If you think this, you're actually wrong. So this statement here is not true. The more genes an organism has, the more complex it will be. That's not true, because in that case, it would mean that the rock has the most genes, which he does not. In fact, the most genes is held by this flower. The most DNA is held by this flower, and second most is this fish, and then a human, and then some bacteria. So the total number of DNA that you have does not correspond to how complex the organism is. Okay? It's not like that. So the more genes you have, the more DNA you have, that does not necessarily mean that you are a more complex organism. Okay? Because we can see that. A Homo sapien, genome size, so genome size means the total number of nucleotide base pairs. You know the letters A, T, C, and G that your DNA is made of. The more numbers of that you have, so the longer your sequence, the size of your sequence. So if you have like many A, T, G, C, C, Cs, that would be a very long genome or a large genome. Whereas if you have few of those letters, that is a smaller genome size. So in this case, we can see a Homo sapien has 3.2 billion letters in a row, whereas this flower actually has many times more. It has 150 billion of those letters in a row. So you can see it has far more DNA than us, but we are still more complicated. And the reason being for that is because a lot of our DNA is non-coding DNA, meaning it doesn't mean anything. Okay? So it's there, but it doesn't do anything. So that just means humans have less non-coding DNA. We make use of most of our DNA, unlike these organisms. So just a big takeaway is just know that the more genes or the more DNA does not mean you are more complex of an organism.